DNA is the primary information repository in a cell, carrying the instructions needed to make cellular products that contribute to the survival and function of a cell. Among these products are various types of RNAs and proteins. RNAs are produced after a transcriptional event, in which the cell exposes a specific section of single-stranded DNA and synthesizes a complementary strand of RNA. Some of these RNAs, including transfer RNA and ribosomal RNA, may undergo some post-transcriptional modifications, but are essentially functional in their current state. The only type of RNA that undergoes a further translational step is messenger RNA, or mRNA, which is complementary to a gene that is protein coding in DNA. In this process, groups of 3 nucleotides on the mRNA strand are read by ribosomes in the cell to ultimately produce functional proteins. This transfer of information from DNA to RNA to protein is summarized by the central dogma of genetics. This video will review the steps of transcription in prokaryotes and eukaryotes, with specific emphasis on their differences. Let's begin with a look at transcription in prokaryotes. The DNA of prokaryotes is arranged in a single circular chromosome that is suspended in the cytosol. Short sections of this chromosome are protein-coding genes. Now, to get from double-stranded DNA to single-stranded RNA, only one strand must be read. But which strand of DNA must act as the template? Well, synthesis by RNA polymerase can only proceed in one direction, and that is 5' to 3', just like synthesis of DNA in DNA replication. And since the strands must be antiparallel to one another, that means that the only strand of DNA that can be read as a template for the synthesis of RNA must be 3' to 5'. The strand of DNA that is read as the template is therefore known as the non-coding or antisense strand, since it is a sequence complementary to what will appear in the transcribed mRNA. And the strand of DNA that is not used as a template is known as the coding or sense strand, since its sequence is equivalent to that which will appear in the mRNA. In this template DNA, there is a consistent structure associated with a transcription unit. There is the actual protein coding sequence that contains instructions for protein synthesis in a series of 3 base codons. But the sequence is flanked on either side by important DNA elements. In front of every gene is a promoter. The promoter is not itself transcribed because it just acts as a recognition site where free-floating RNA polymerase can attach to the DNA in preparation for RNA synthesis. Transcription only begins at the +1 start site. This +1 base is the first base to appear in the transcribed mRNA and can be any of adenine, thymine, cytosine, and guanine. Following the +1 site is a region that is not yet part of the coding sequence. It is a region that will be transcribed but not translated, and act as a recognition site for a ribosome before translation begins. Since this region will be on the 5' end of the mRNA strand, it is known as the 5' untranslated region, or 5' UTR for short. Behind the protein coding sequence is a similar 3' UTR. So it is a series of RNA nucleobases that appears only in the transcribed mRNA, but that will not be translated by a ribosome. Finally, there is a terminator sequence in DNA. This sequence is fully transcribed into mRNA by RNA polymerase, before RNA polymerase recognizes that it must detach from the DNA and stop transcribing. Although it is a regulatory site in DNA, this terminator sequence is not understood as a signal to stop transcription until after it itself has been transcribed into RNA. The process of transcription begins with an initiation step. The initiation step is dependent on the structure of the promoter region. The promoter is largely an assembly of random nucleobases with no specific purpose or sequence except for in two distinct regions, the -10 and -35 regions. These regions are referenced by their distance in nucleotides from the +1 start site. Therefore, the -10 region is approximately 10 bases away from the +1 start site, and the -35 region is approximately 35 bases away. Now, the key thing about the -10 and -35 regions is that they are consensus sequences. What this just means is that the sequences have been really well conserved over time, such that geneticists can predict what bases are most likely to appear in these regions. Of course, not every promoter in prokaryotes of a given species will have perfectly identical -10 and -35 sequences, but all the promoters will have some degree of resemblance. For instance, in this image, we compare the promoter sequences of many genes in a given bacterial cell. The first thing we might notice is that these promoter sequences are not perfectly aligned, but the -10 and -35 distinctions are just approximations. The second thing we should notice is that not all the -10 regions are identical with one another, and not all the -35 regions are identical with one another. But what we are able to do is consider this whole long list of promoters and define a new consensus sequence that includes only the most frequently observed bases at each position in the -10 and -35 regions. How closely a given promoter matches the consensus sequence tells us how strong that promoter is. The more bases that do not match the consensus sequence, the weaker the promoter. And this has implications for how readily RNA polymerase is able to bind to the promoter to initiate transcription. Because that's what a promoter is used for, the binding of RNA polymerase to DNA. RNA polymerase is the enzyme that will be used to synthesize mRNA. It is composed of a couple different subunits, one of which specifically recognizes the -10, -35 regions of a promoter for binding. Note also that the -10 region is mostly composed of thymines and adenines. How can we rationalize this observation? Well, DNA exists in a double-stranded state in the cell, but it can only be transcribed once the two complementary strands of DNA have been separated. RNA polymerase is the enzyme that separates these strands during transcription, and it does so beginning at the -10 region, because it's easier to melt the DNA apart in AT-rich regions, where there are only 2 hydrogen bonds between complementary bases instead of 3. After binding, breaking apart the DNA, and scanning the promoter, transcription officially begins at the +1 start site. Step 2 of transcription is elongation. This describes the actual synthesis of mRNA using DNA as a template. RNA synthesis always occurs in a 5' to 3' direction, similarly to DNA replication. But unlike DNA replication, transcription requires no primer. Synthesis begins and completes with RNA, and it is only the synthesis of DNA that requires a primer. Nucleoside triphosphates free floating in the cytosol are the substrates for this reaction. They are grabbed by RNA polymerase and added in an order that is complementary to the DNA template. As an anabolic process, or one that involves the synthesis of large polymers from small subunits, the synthesis of mRNA is endergonic, requiring energy. This energy comes from the cleavage of 2 phosphates, converting a nucleoside triphosphate into a nucleoside monophosphate. As the RNA is being synthesized, there is a region where the RNA is bound to DNA via hydrogen bonds, forming an RNA-DNA hybrid. but the rest of the synthesized RNA strand is disassociated from DNA and trails out of the transcription bubble. Specifically, it is the 5' end of the RNA that trails out like a tail. Multiple RNA polymerases may be operating on the same prokaryotic gene simultaneously. We can determine the directionality of each mRNA and the DNA using the lengths of the mRNAs. Each tail end is a 5' end, since this is the end that must have been synthesized by each RNA polymerase first. The 3' end of the RNA is still within the transcription bubbles. The longest RNA tail indicates which one has been in the elongation process the longest. And since RNA is synthesized anti-parallel to DNA, and therefore the template is read 3' to 5', the longest RNA tail will be closest to the 5' end of DNA. Now, what exactly is the purpose of producing an mRNA intermediate? If mRNA is a perfectly complementary copy of a DNA gene, and therefore does not provide any information that DNA does not already tell us, then why produce it at all? Well, the mRNA intermediate gives the cell the ability to regulate how much protein is being made. By controlling when specific mRNA are being produced, how frequently they are being made, and how quickly they are degraded, the cell regulates the total amount of RNA present in the cell. The cell is not able to exert the same level of control directly at the DNA level. After all, we cannot simply change the amount of DNA in the cell. We need all of it present at all times. Therefore, mRNA offers the cell lots of opportunities for control and regulation of protein synthesis, and this is why the mRNA intermediate is so important, even if it increases the length of time required for protein synthesis. Finally, RNA polymerase reaches the terminator sequence, and the termination stage of transcription begins. The important thing to note about the terminator sequence is that it cannot be recognized by RNA polymerase in the DNA form. The entire terminator sequence is first transcribed into RNA, which in many cases has the unique property of being able to immediately fold into a hairpin loop. This hairpin loop is able to form whenever the terminator sequence is an inverted repeat, which essentially allows the terminator sequence to form self-complementary base pairing. The formation of the hairpin loop is assigned to RNA polymerase to disassociate. Alternatively, a specific type of protein called the Rho protein aids in the detachment of RNA polymerase from DNA and the termination of transcription. The actual dissociation of RNA polymerase from the DNA is facilitated by another AT-rich region following the terminator sequence. Again, as we saw for the initiation step, this is because of a predominance of only 2 hydrogen bonds between complementary bases of DNA as opposed to 3. The resulting mRNA contains the 5' UTR, the protein coding sequence, and the 3' UTR, and is immediately ready for translation. When an mRNA contains only one protein coding sequence within it, it is considered a monocistronic RNA. But in prokaryotes, it is possible for up to 3 and even 5 protein coding sequences to be grouped together under the control of one promoter. This just means that when RNA polymerase binds to the promoter to initiate transcription, it will transcribe all protein coding sequences at once into one single mRNA, which we call a polycistronic mRNA. This configuration is called an operon, and it increases the efficiency of protein synthesis, particularly because the protein coding genes grouped together under a promoter are all involved in the same metabolic pathway. For eukaryotes, only monocistronic RNAs are produced, because each protein coding gene is singly placed behind a promoter. Eukaryotes are not observed to group protein coding genes together into operons. Aside from this, there are a couple other differences between transcription in eukaryotes and transcription in prokaryotes, most of which occur during the initiation and termination stages. In eukaryotes, initiation also begins at the promoter, but there are three distinct differences between prokaryotes and eukaryotes. Difference number 1. Instead of having the -10, -35 recognition sequences seen in prokaryotes, eukaryotes have just one recognition sequence called the Tata box. This Tata box is so named because it is a short sequence of thymines and adenines. Difference number 2. While RNA polymerase is able to bind directly to the DNA of prokaryotic promoters, transcriptional factors are needed to first bind to the Tata box in eukaryotes, and only then can RNA polymerase be recruited to the DNA. There is no direct recognition of the promoter by RNA polymerase in eukaryotes. It is the transcriptional factors that are key mediators of this interaction. Difference number 3. While prokaryotes regulate rates of transcription directly at the promoter, eukaryotes tend to regulate rates of transcription at sites more distant from the promoter, sometimes even hundreds to thousands of bases away. These distant regulatory sites are bound by activator and repressor molecules, which are sometimes proteins and sometimes metabolites, and can either increase or decrease rates of transcription, offering the cell greater sensitivity to cellular conditions. But just like in prokaryotes, once RNA polymerase is recruited to the DNA, it will scan the DNA and start transcribing at the +1 start site. Ribonucleoside triphosphates are again added into the growing strand of RNA during the elongation stage, and it is the liberation of 2 phosphates that provides the energy to do so. During termination, there is again a difference. Instead of transcribing a terminator sequence, which quickly forms a hairpin loop, as prokaryotes do, eukaryotes will transcribe right through what we call a cleavage site. Only after the cleavage site is transcribed into RNA is it recognized by an RNase enzyme, which will cut the mRNA strand away from RNA polymerase. This cut also signals to RNA polymerase that it must stop transcribing. Therefore, termination of transcription is dependent on an external RNase protein. There are 2 other major distinctions between prokaryotes and eukaryotes that are not directly related to the action of RNA polymerase. The first major distinction is the presence of a nuclear envelope around DNA in eukaryotes. Because of this nuclear envelope, there is a physical barrier separating the location where transcription occurs and the location where translation occurs. Furthermore, the cell needs some mechanism to export mRNA made in the nucleus out into the cytosol, where the ribosomes are found. In prokaryotes, the absence of a nuclear envelope means that transcription and translation can occur simultaneously, and this is observed to occur as soon as the 5' end of mRNA exits the transcription bubble. The second major distinction is the structure of the protein coding sequence. In prokaryotes, the protein coding sequence in DNA perfectly describes the sequence of amino acids needed in the final protein product for it to be functional. Therefore, the mRNA produced in prokaryotes is ready to be immediately translated. But in eukaryotes, the protein coding sequence is a mixture of introns and exons. Only the exons must be expressed, and the introns must be cut away from the mRNA before it is ready to be translated. Therefore, mRNA produced after transcription is only considered prerequisite mRNA or pre-mRNA in eukaryotes. And this pre-mRNA must undergo post-transcriptional modifications before becoming mature mRNA that is ready to be exported from the nucleus and translated. In my next video, I will review mRNA processing and alternative splicing, which enables a eukaryote to maximize the usefulness of a given protein coding sequence to produce a family of related functional proteins.